Okay, welcome everybody. Well, I thought today I would just continue from last week where we started discussing the RTAs. And for those of you that weren't there, I'll repeat some of the concepts and details that we discussed. So just to review before we get into the clinical, the kidney has to perform two essential roles to keep the blood bicarbonate at a level of about 25 millimolar. The first is that it has to reclaim the bicarbonate that comes through the glomerular filtrate. Remember that roughly a box of Arm & Hammer baking soda comes through the glomerulus every day, and if you don't reclaim that, your blood bicarbonate is going to get very low just because it's going to be excreted in your urine. So that's an essential role of the kidney to absorb the filtered bicarbonate. But that's not enough. Even if the kidney was able to absorb all the bicarbonate coming through the glomeruli, there's another process which also tends to decrease the blood bicarbonate concentration, and that's the fact that because of the food we eat, our liver generates hydrochloric and sulfuric acid every day, and the protons that are derived from those two acids would deplete bicarbonate. Even if we absorbed all the filtered bicarbonate, it's like you have a proton IV, a hydrochloric acid and sulfuric acid IV, except the hepatic vein is where the protons are coming into the IVC. So something else has to occur in order for the blood bicarbonate to not decrease because of this process. And what the kidney does is it generates additional, or what we call new bicarbonate, above the bicarbonate that it reclaimed through the glomerular filtrate. So if either of these processes is defective, you'll end up with a metabolic acidosis. So let's just review the mechanism by which the kidney reclaims the bicarbonate that comes through the glomerular filtrate. And it's mostly the proximal tubule. So that's really what I'm going to discuss today. It absorbs approximately 75 to 80% of the bicarbonate coming through the glomeruli. And I'm not going to discuss what the rest of the nephron does today. And defects in proximal tubule bicarbonate absorption are responsible for most of the defective bicarbonate absorption in the kidney. And that's why when you have defective bicarbonate absorption, it's usually due to the proximal tubule, and that's why we call it proximal RTA. We don't have RTAs that are due to abnormal loop of Henle bicarbonate absorption or defective collecting duct bicarbonate absorption, because quantitatively they're just not important enough, nor have clinical syndromes that involve those segments been described pathophysiologically. So when you have defective bicarbonate absorption, you're really talking about, in the nephron and the whole nephron, you're really talking about a proximal tubule functional abnormality, not anywhere else in the nephron. So let's again review how bicarbonate gets reabsorbed. And remember, the important concept is it doesn't get absorbed directly. It's an indirect process. And the first thing that has to happen is protons have to be secreted by the proximal tubule cell into the glomerular filtrate. And there's two mechanisms for doing that. The major mechanism is via a protein called NHE3. NHE3 stands for sodium hydrogen exchanger 3. And this protein is one of 8 different sodium hydrogen exchangers in the mammalian genome. All mammals have NHE3 in the apical membrane, not other types. They're related by their amino acid sequence, so they belong to this so-called NHE family. And they're throughout our body in different cell types. But the one that's responsible for reclaiming bicarbonate is the third version, NHE3. So this protein transports one sodium ion into the cell in exchange for a proton. There's another mechanism for proton secretion that we'll talk about more as it's related to distal RTA or type 1 RTA, and that's what's called a vacuolar proton pump. And it's shown here as just an H+. It's actually made up of 14 different proteins. It's a very complicated molecular machine, and it's also in the proximal tubule. It's a minor component of proton secretion in the proximal tubule. So these two mechanisms account for the proton secretion. And what then happens is the protons that get into the lumen bind to the filtered bicarbonate, shown here as HCO3, and convert the HCO3 into water and carbon dioxide. So then what happens is that the carbon dioxide and the water go across the proximal tubule apical membrane. This is called the apical membrane on this side. They don't cross the lipid bilayer. They don't cross the apical membrane per se, as far as the lipid component. They actually go through a protein that's inserted in the membrane called aquaporin 1. And you might recall aquaporins are proteins that allow water to go through. They also let gases to go through, like carbon dioxide, which is not as well known. But the key is that the carbon on the bicarbonate here gets through the apical membrane as carbon dioxide. So once the carbon dioxide gets into the cell, it recombines with water to make carbonic acid, which then breaks up into two protons and carbonate. Both this reaction and the reverse reaction are catalyzed by carbonic anhydrases. The one in the lumen is catalyzed by carbonic anhydrase 4, which is actually attached to the apical membrane via what's called a GP1 anchor. The carbonic anhydrase 2 in the inside the cell is floating around freely in the cytoplasm. And they don't do anything other than accelerate these processes. They don't change the process. They just make it occur faster, more efficiently. So once the carbonate is formed, it gets out of the cell on another protein called NBCE1A that we've studied for a number of years in my laboratory. And this protein transports one sodium ion and a carbonate. And what you can see is that the charges on these two things are not equal. So this transport process actually carries what we call an electrical current across the membrane here. And because it transports a current, it actually senses the voltage here on the membrane. That is always here due to the function of the sodium potassium ATPase and potassium channels here, which are on the basolateral membrane of the proximal tubule cell. And it's actually the voltage here that actually drives the transport from the cell to the blood side. So this is the normal set of transport machinery that allows the bicarbonate that came through the glomerulus to end up in the renal vein. It's a very indirect process requiring a lot of different steps and a lot of different proteins. And there's nothing unique to the human kidney. It's present in all mammalian kidneys. So that's what must occur in order to not get proximal RTA. Now, the second thing I mentioned is the kidney must generate new bicarbonate above that amount to match the acid that the liver is pumping in to the hepatic vein every day because of the type of food we eat. So again, we have this proton production by the liver, and it's around 50 milliequivalents a day of protons in the form of hydrochloric and sulfuric acid. I didn't show the anions here. And the kidney generates an equal amount of new bicarbonate. This is not the reabsorbed bicarbonate. This is the additional amount of new bicarbonate that's been generated that matches the daily hepatic proton load, and you end up making carbon dioxide, which in a net sense, the red here is your whole body shown in a simplistic way. You end up generating carbon dioxide from this reaction, which your lungs just excrete. So this is occurring in all of us every day. Now, how does the kidney make new bicarbonate? Well, the major source is from the amino acid glutamine. And the glutamine is converted, not shown here, but this occurs in the mitochondria, to a substance called alpha-ketoglutarate, which has two carboxyl groups on it. When alpha-ketoglutarate enters the Krebs cycle, it's converted into two bicarbonate ions and also two ammonium ions. So this is the major source of new bicarbonate generation by the kidney that helps it match the daily proton load. Now, the other thing the kidney must do is not allow the ammonia that it made as a side product of this reaction to get back into the blood. If it gets back into the blood, then you're just going to take the 2 bicarbonate and 2 ammonium ions and convert this back to urea in the liver. Urea is made of 2 bicarbonate and 2 ammonium ions when you look at the net cycle of the urea cycle. So it's important that the kidney tries to not send this ammonia back into the renal vein. Of course, all the new bicarbonate is sent into the renal vein via NBCE1A. It's not sent into the urine. And we'll talk about that because the kidney doesn't actually send all the ammonia back into the renal vein. Some of it goes into the urine, which we know. We measure the urine anion gap or the urine osmolar gap, or we measure the ammonia directly. It's coming from this reaction predominantly. The other thing that must occur is the kidney needs to convert HPO4 2- that's in the lumen to H2PO4. Every time it does that, it generates 1 new bicarbonate ion. And I'll show that process also. So all these 3 things must occur in order for the kidney to generate new bicarbonate to match the daily hepatic proton load. This has nothing to do with bicarbonate reclamation from the glomerular filtrate that we talked about initially. So as far as the new renal bicarbonate generation from alpha-ketoglutarate, it comes from glutamine. The glutamine gets into the proximal tubule cell, and in the mitochondria is converted to 2 bicarbonate ions, which is sent via NBCE1A that I showed before into the renal vein. So NBCE1A transports not only the bicarbonate that came through the glomerular filtrate, but also the new bicarbonates that generated. And it's actually transporting carbonate, as I showed, not bicarbonate, but simplistically here it's shown as bicarbonate. But in this reaction in the mitochondria, 2 ammonium ions are made. And it's important, as we'll talk about in more detail, that this, as much as possible, this ammonium does not get back into the renal vein like the bicarbonate did. So shown here is a concept called renal ammonium partitioning. The proximal tubule makes the new bicarbonate, as I mentioned, and that new bicarbonate is sent via NBCE1A back to the renal vein. So that's good because now you would think you'd have all this new bicarbonate to match the daily hepatic proton load. However, the ammonium that's made in humans differs in different animals, but in humans don't send all this ammonium into the urine as one might think would be advisable, because then you would free up all this 100% bicarbonate that's generated to be free to match the daily hepatic proton load. It turns out in humans, when the renal vein and urine ammonia delivery was measured years ago in humans, it was found that about 60% of the ammonia that the human kidney makes is sent to the urine, which is good. But not all of it is. 40% actually goes back to the renal vein, and that's not a good thing because it's using up 40% of this new bicarbonate and urea is generated. So basically, you've taken 40% of the glutamine and turned it into urea, which is fine as far as nitrogen balance, but it's not fine as far as freeing up 100% of the bicarbonate to match the daily hepatic proton load. But it just turns out that there's extra amount made here, so there's sufficient amount left over to match the daily hepatic proton load. Plus, under different conditions, and I'll talk about some of them, the kidney actually modulates this ammonium partitioning concept, changing the percent of the ammonia that's made, how much of it will be delivered to the renal vein or the urine based on the different acid-base conditions that the patient is facing. And we'll talk about that in more detail a little bit later. Now, the ammonia that's made in the proximal tubule goes through a number of steps not shown here in the nephron, but finally ends up as NH4 plus in the urine. It actually gets into the collecting duct through these 2 proteins called RhCG and RhBG. These are Rh proteins that you're familiar with red cell hemolysis. You know, the Rh factor. Well, the same proteins are in other cells, and they're very important. They're actually ammonia transporters. That's what they do in the red cell also. That's not well known. But so they're important for letting NH3 from outside the collecting duct into the lumen. And then the conversion of NH3 to NH4 plus occurs via the same protein that we talked about earlier, a protein complex. Remember, this represents 14 different proteins. The vacuolar proton pump, which is responsible for acidifying your urine and urine in the collecting duct. When you say your urine pH is 5 or 5.5, it's because of this complex of proteins called the vacuolar proton pump. And we'll talk about that a little more in detail in a little while, because there are mutations in this protein that cause distal RTA. So it's very important not only to generate the bicarbonate, but to have these processes in place to assign the correct amount of ammonia into the urine versus the renal vein. Because remember, any ammonia getting back into the renal vein, you're just using up the bicarbonate that you generated to make urea, and you lose that bicarbonate as a source of matching your daily hepatic proton load. The other thing that must happen to generate enough new bicarbonate is the conversion of HPO4 to HPO4. It occurs in the proximal tubule and the collecting duct, and the carbonic acid that's in the cell is the source of the proton. So every time a carbonic acid splits and secretes a proton, one new bicarbonate ion is generated, and the cells have different transport processes. I mentioned NBCE1A in the proximal tubule. We'll talk about what happens in the collecting duct, which does not express NBCE1A. It's via another protein called AE1, which is also mutated in distal RTA, and we'll show that a little bit later. So the new bicarbonate generation from this process occurs not only in the collecting duct, but also the proximal tubule. And the correct terminology for it is titratable acid excretion. So getting back now to the clinical diseases, recall that there's a number of different types of RTA. They're not in order as far as the Roman numerals. They're more anatomical here, where proximal RTA is type 2. And there's no logic for this. It's all due to Morris and McSherry and Sebastian. The references are there if you want to read them. This is where the typing first occurred in the medical literature. So there's type 2, which is the same thing as proximal RTA, distal RTA, or type 1. Type 3 is a combination of type 1 and 2. And type 4 really refers to hyperkalemic RTAs that are all distal. There's no hyperkalemic proximal RTAs. So type 4 is again a distal RTA, but it's distinguished from these other RTAs in that they tend to be hypokalemic. And we'll talk about the mechanism for the hypokalemia too, which is still not well understood. It's one of the items in nephrology that have not been worked out. Why these patients with distal RTA are hypokalemic is a very complicated answer, and nobody knows why, in all honesty. And these can be distinguished as to whether they're occurring on a genetic basis or whether they're acquired, like all things in medicine and nephrology. So what I'll continue to talk about is the proximal and distal RTAs of the hypokalemic type that are genetic as far as their basis. So here's a 57-year-old female. I showed this last week. For those of you who have seen it, it's just a repeat. For those of you that weren't here last week, this would be new, but it's a patient who, based on the clinical situation for your exams, you're going to have to be able to tell what this is, because there is really only one cause of this in medicine once you recognize the pattern. So it's someone who had blindness, bilateral glaucoma, and cataracts. And on exam, she had short stature, and this is a real patient, by the way. Who on exam, she had bilateral corneal opacities, abnormal teeth, and an intellectual disability. And so this was her eye. It showed so-called band keratopathy, or if you want, corneal opacities. The teeth were abnormal, and she had calcification of the basal ganglia. And these were her blood chemistries. So she had a non-GAP, or a hyperchloremic, if you want, metabolic acidosis. We don't know if it was compensated or not properly, because we don't have the blood gas PCO2 listed here. And the K was pretty normal, although you have to think that maybe it would have been, maybe it's been artificially elevated because of the acidemia. Remember, metabolic acidosis causes a translocation of potassium out of the cells. And if the pH had been normal here, the K might have been low or lower than this. So you have to be careful in first thinking, oh, the patient's normal kalemic. Yeah, it's normal kalemic based on what you're used to seeing, but it's really not what you would expect to see with this pH. If this patient has started off with a K of 4 with this pH, the K might be 4.8 or 5 even. So having a K of 3.5 with a pH of 7.27 is not hypokalemia in an absolute sense, but you have to start thinking that maybe there would be hypokalemia here pathophysiologically had the pH been normal. I'm sorry, here the PCO2 is here. I didn't see it. So the PCO2 is 27 with a bicarb of 12. Those changes from normality are about equal. So this is what you would expect in a compensated metabolic acidosis. So there's no mixed disturbance occurring here. And the urine pH is 4.9. So as you get more experience from the clinical scenario, both those pictures and what is shown here, this can only be proximal RTA on a genetic basis. There's nothing else in medicine that causes this phenomenology. So that would be one reason for wanting to work the patient up for proximal RTA further. Plus, proximal RTA is one of the causes of a non-GAP metabolic acidosis with a low urine pH. Now, there's a subtlety here, and we'll talk about that in a minute, in that the urine pH, and that surprises certain people. I mean, how can you, the obvious thought is, how can you have a low urine pH when you can't absorb bicarbonate normally? And the answer is, and we'll talk about that in a minute, there's two phases, or sort of two extremes to what you'll find in the urine with these patients. And we'll talk about what those two extremes are in a minute. But if you do suspect proximal RTA, the first thing you order on the chart is a fractional excretion of bicarbonate. That's how the diagnosis is made objectively. So in this patient, the fractional excretion was 30%. Remember, the normal is about 1% or less. We don't excrete 30% of the bicarbonate coming through our glomeruli. And remember, like the fractional excretion of anything, this is how you plug the numbers in. You get a spot urine and get these numbers. You can also get the total CO2 if you want. You can send the blood off, the urine off for chem panel, instead of getting blood gases and getting a calculated bicarbonate. So either way, you plug in either the total CO2, or if you happen to get blood gases from the urine, it's called blood gases. You have to order it like that because they're not used to seeing urine gases. But be that as it may, you plug in either the calculated bicarbonate from the gases in the urine or the total CO2 from your chemistries that you've gotten from the urine. And you'll get this number when you multiply by 100%. And as I said, normally it should be 1% or less. So this patient had isolated proximal renal tubular acidosis. It's now known to be autosomal recessive. It's due to mutations in NBCE1A. The protein cannot transport sodium and carbonate across the basolateral membrane out of the cell. So the whole system just backs up. It's in this portion of the nephron here with the flashing green square, where the majority of bicarbonate, or if you want to say carbonate, that's also fine, is absorbed in the nephron. And there was no other phenomenology. The rest of this is a scanning EM of the proximal tubule. This is looking at it from the lumen just after the glomerulus. This is the apical membrane where NHE3 is, and where the GPI anchored carbonic anhydrase 4 is. This is the basolateral membrane where NBCE1A is located, the K channel that I alluded to, and the sodium pump. This is the inside of the cell where carbonic anhydrase 2 is floating around. So bicarbonate can't get vectorially across this entire pathway, and you just end up dumping it all in the urine. And we'll talk about why the urine pH in the patient is low and not high in just a sec. So again, this patient and others in the literature have mutations in NBCE1A. This is just a picture of what the protein actually looks like from our lab that we published in 2018. There's two components here. They're both identical. They're called monomers. A lot of these proteins are what are called dimers, where two subunits bind to each other, and that's what you see with this transporter. Also, the sodium and the carbonate actually go through the middle of each of these. So this would be the outside of the cell, and this is the inside, the cytoplasmic side. So the sodium and carbonate are actually coming out from inside the cell and going to the renal vein this way. And there are mutations throughout the transporter that lead to the phenotype of the clinical disorder. But this protein, NBCE1, is not only in the kidney, it's in the brain, and we won't get into all the papers that have discussed what it's doing in the brain. But when it's not functioning, patients can end up with this phenotype. It's also very important to keep the cornea in your eye allowing photons through to the rest of the eye. When it's not functioning properly, you end up with band keratopathy or cloudiness in your cornea. It's also important to maintain intraocular pressure normally, and also to prevent cataracts. It's actually in the epithelial cells surrounding the lens in our eye. And each of these has a whole story in and of itself that there's just not enough time to get into today. But the whole point is that this transporter has different functions throughout the body, and that's why the patients have an extrarenal phenotype that you need to memorize in order to recognize immediately whether this is the transport protein that's mutated or involved in your patient. It's also essential to make the enamel in our teeth. When it's mutated, your teeth become abnormal and are not covered with normal enamel, as you saw in the picture of the patient earlier. Now, getting into why we have two different phases of what the urine pH is, again, the cause of the metabolic acidosis, as I mentioned, was defective bicarbonate absorption, and that is sort of easy to understand. If you don't absorb the bicarbonate, it's going to come out in the urine, and your blood bicarbonate is going to fall, which is the cause of the metabolic acidosis. But then why do we have two different types of phenomenology for the urine pH? And the reason is there's a temporal effect. When the process was first created, you're going to dump a ton of bicarbonate in the urine. So if you have someone who's just put on acetazolamide or ifosfamide or something that blocks the process in the proximal tubule, you'll suddenly get a flood of bicarbonate coming through the rest of the nephron, and the rest of the nephron doesn't have the transport machinery to reclaim the bicarbonate, and so it ends up in the urine. And there you'll find actually a high urine pH. But the key point is, and that's shown here pictorially in this cartoon, where you punch a bunch of holes in the bucket here, the water representing bicarbonate is going to start coming out onto the floor, and you'll find a lot of water on the floor, or as an analogy, a high urine pH. But once the bicarbonate, but that then leads to a lower bicarbonate in the glomerular filtrate. As the metabolic acidosis gets worse and worse, you're going to have less bicarbonate coming through the glomeruli, just to a point where the defective bicarbonate reclamation in the urine can now handle that significantly lower load coming through the glomeruli. And once the rest of the nephron, including the proximal tubule, can handle that newer, significantly lower load, the urine then won't have bicarbonate in it anymore, and the urine pH will then be able to become acidic. It becomes more acidic than normal because the collecting duct actually tries to compensate by revving up its proton secretion. So if you look at patients with proximal RTA, they actually have a very high collecting duct acidification going on. So you end up with a pH that is even lower than normal once you've achieved the ability to reclaim the new significantly lower filtered load. So that's a trick that people don't always get on an exam. And so since in patients with genetic disorders, they've had it from birth, you're always going to be in a steady state unless the patient is given bicarbonate, which makes things more complicated. But if you just have a patient who's untreated, by the time the pediatric nephrologist sees the kid, the urine pH is going to be low because they've already dumped all the bicarbonate. They're able now to reclaim what's coming through. the new lower load. And it's only when they're given bicarbonate or given citrate that the urine pH will become high again. But that's just because, again, there's more bicarbonate coming through the glomeruli than the nephron can absorb. In contrast, if you're given acetazolamide and you happen to be measuring the urine pH in a patient before and after, you will be able to see the urine pH become increased above what it was prior to the acetazolamide. And if you keep the acetazolamide on board and don't treat the patient, or Diamox for those that are more familiar with that term, if you keep giving the patient Diamox, the urine pH will come down after, again, you've dumped all that extra bicarbonate. So again, there's going to be this temporal effect on the urine pH that you really need to be aware of, or you're going to get confused and you're going to miss diagnosis, depending on what time frame you're looking at. Now, there's a second mechanism for metabolic acidosis and proximal RTA, which is really not well known by most people, but you just have to keep that in mind. So the lack of reclaiming bicarbonate it makes sense, but there's an additional factor, and that is, and it's not well understood why, but there's a suggestion that the glutamine conversion to alpha-ketoglutarate might also be defective. Now, why that is, people have some evidence for in animals and in some humans, and the suggestion is maybe the cell pH is lower than normal also, not just the blood, but the cells also have abnormal pH, and that's impairing these processes. And so if you don't make enough new bicarbonate from alpha-ketoglutarate, then you're not going to be able to match your daily hepatic proton load as easily. So that's another mechanism that people have talked about. But the major mechanism is an abnormal absorption of bicarbonate from the glomerular filtrate, not this process, which involves new bicarbonate generation. It's not the major cause. So here's another patient. Again, it's a 3-year-old female. This is a real case with decreased hearing and polyuria. She was 18 percentile for height. She had bilateral sensory neurohearing loss and rickets. Again, once you get more used to these things, you'll have the diagnosis right away, even without getting any chemistries from the patient. And the blood showed hypokalemia with a metabolic acidosis, non-GAP, and again, it was compensated appropriately. So this is a hyperchloremic or non-anion GAP metabolic acidosis. And when the urine pH was measured, it was 7.1, and the patient was also excreting a lot of calcium. And the x-rays showed nephrocalcinosis, which again, remember, is calcification, not in the lumen of the tubules, but the interstitium. Okay, so once you get used to it, this really has only one diagnosis. But for those that are not used to seeing these patients, we'll go a little further. So this diagnosis, just from the chemistries, I'm not talking about the phenotype, but just from the chemistries and the hypercalciuria and the nephrocalcinosis, can only be hypokalemic distal RTA, hypokalemic type 1 RTA, or if you want, type 1 RTA, because they're never hyperkalemic. And this is where the phenomenology is occurring. It's occurring here in this portion of the collecting duct, and it involves a cell called the type A intercalated cell. The chordal collecting duct has 2 cell types. This is the intercalated cells, which have these worm-like projections. You're actually taking a picture from inside the tubule. So you're sitting in the urine side. This is not from outside the nephron, it's from inside the nephron. So you're looking at the apical membrane. And if you just picture the camera sitting in the urine, that's what you're looking at. You're looking at the surface of the cell facing the urine. These worm-like projections, as I will talk about in a minute, have a ton of proton pumps on them. And you can immediately distinguish these cells from the principal cells here. These principal cells are more polygonal, not circular. And this white thing here is actually a central cilium. And people have thought for a number of years, this is actually a flow transducer. It actually moves in different orientations as the urine flows along here, and actually is transduced into changes in calcium inside the cell that alter the cell's transport properties. This is the cell that responds to aldose. So when you give someone aldo and you get more K in the urine, it's this cell that's secreting the potassium. This is the cell that also has the ENaC sodium channel that is blocked with a miloride. So when you give a miloride, you're actually impairing the sodium absorption in this cell. And also, it's the cell involved in Little syndrome that we talked about a few weeks ago. So this is the anatomy of the cortical collecting duct, but the problem with these patients is in these intercalated cells sitting here. So we'll look at that cell in a little more detail. It's called the type A intercalated cell. And this is the molecular machinery that allows it to acidify our urine. And it is responsible for the pH that we have in our urine whenever you're measuring a patient's urine pH. So protons are secreted not via NHE3, but via this proton ATPase. And I'll show a more three-dimensional view of that in a minute. There's also a more minor transporter called an HKTPase here. It's just shown for completeness. It's more important in the intestine. There is carbonic anhydrase 2 floating around here. The protons come from carbonic acid, not shown here, but break up into proton and bicarbonate. And then the bicarbonate, and not carbonate, but bicarbonate leaves the cell on another protein, not MBCE1A, but a protein called AE1. AE stands for anion exchanger because it exchanges a bicarbonate for a chloride ion. This is actually in red cells and makes up 60% of all the protein in our red cells. And it's mutated in different morphologic abnormalities in red cells, as I'll talk about in a minute, such as spherocytosis and elliptocytosis. But it's the key protein responsible for bicarbonate transport across the basolateral membrane into the renal vein in the collecting duct. And there's another protein here called CLCKB. You might remember this is one of the proteins that's also in the thick ascending limb that's mutated in Bartter syndrome. I just show it again here for completeness. So patients with isolated distal RTA on a genetic basis are now known for a number of years to have mutations in the vacuolar proton ATPase, and that's why they can't acidify their urine. So this is what this protein complex looks like, and there are actually 14 different proteins that make up the vacuolar ATPase. It's divided into two subunits, a V1 subunit, which is the green here, and a V0 subunit. The V0 actually sits in the lipid bilayer. The V1 is in the cytoplasm. So if you imagine an inverted cell here with the cytoplasm here, this is the lumen where the H is of the collecting duct. This is the urine side. This whole complex here is responsible for splitting ATP. These proteins are responsible for translocating a proton from the cell into the lumen. The B1 subunit has been shown to be mutated in these patients, and they end up with this phenotype: hypokalemia, nephrocalcinosis, hypercalciuria, calcium phosphate stones, hypocitraturia, decreased urine ammonium, elevated urine pH. But because the transporter is also important in the inner ear, these kids get deafness. In fact, a number of proteins or transport proteins in the kidney or in the inner ear too. There's a whole bunch of diseases where you get inner ear kidney phenotypes. This is one of them. And they also get rickets and bone abnormalities, possibly from the pH per se in the blood, but also this protein is involved in bone cell phenomenology also. For instance, it's very important in osteoclasts. Okay, so different organs involved, again, a multi-organ disease, and not just, as you see here, a problem with pH. All these different transport processes, the hypokalemia, the hypercalciuria, the stones, there's more than just acid-base abnormalities in these patients. We'll talk about that in a second. In other patients, they have the A4 subunit that's defective. And again, that's in the V0 subunit, which is made of 6 different proteins. The V1 is made up of 8 different proteins. So there's 14 total. Again, these patients have a similar phenotype. including the sensory neural hearing abnormalities. Now, in addition to the vacuolar ATPase mutations in patients with genetics forms of distal RTA, AE1 also is known to be mutated. So the protons can be secreted, but bicarbonate can't get out of the cell in these patients. They end up with a similar phenotype. There's two forms, a recessive form, which has been described in the tropics, where it's more prevalent and quite severe phenotype as far as the acidemia, hypokalemia, nephrocalcinosis, bone abnormalities. As I mentioned, 60% of the red cell membrane protein is AE1, and so they also get red cell abnormalities. In the dominant form, it's more prevalent in Caucasians. That would be the type you'd see at UCLA if you ever had a case. It's rare, and the phenotype is less severe. And that's just due to the different mutations that you get. You get different types of mutations in the transporter than in the recessive form, which affect its function more severely. A little more recently, there's another transcription factor called FOX11, and mutations in FOX11 are also known to cause distal RTA of the hypokalemic variety with sensory neural hearing abnormalities. This is a transcription factor. It's not involving a transport protein per se, but the expression level of the proton pump subunits and also AE1 are affected in these patients. So it affects multiple proteins in the transport pathway in the intercalated type A intercalated cell. Now, there's something else called incomplete distal RTA that you may have come across. Incomplete means that the blood pH and bicarbonate are normal, and yet they still have what's called distal RTA. They tend to have normal systemic acid-base numbers, but they have a low citrate excretion in the urine. They can get nephrocalcinosis, they can get phosphates, calcium phosphate stones, and they get hypercalciuria. I should mention about the stones that was mentioned before. These patients don't get calcium oxalate stones. So if you get a calcium phosphate stone in a patient, you have to think potentially of RTA, distal RTA. Now, in the literature in the 1950s, people gave people ammonium chloride to acidify their blood. Remember, as I mentioned, it acidifies the blood not because the NH4 deprotonates. The pK is about 9.3. The NH4 enters the hepatocytes and consumes bicarbonate and makes urea. That's why the hepatic venous blood drops its pH. The bicarbonate concentration drops, and then that acidifies the entire IVC and systemic blood. So it's a way of acidifying the blood. You just give a patient ammonium chloride tablets, or normal person ammonium chloride tablets, and you can make their blood pH whatever you want just by giving more or less ammonium chloride. And they found that the urine pH was elevated, despite the fact that they had no systemic abnormalities. A normal person should acidify down to a certain level, usually less than 5.5. These patients don't. And so they have a subtle acidification defect that was not severe enough to change their systemic acid-base balance, which is why it's normal. So you can only pick up the subtle acidification abnormality by giving them ammonium chloride. That was the thinking. Now, since then, people have looked at this in more detail and now break up, and you'll need to know this for your exam. You break up incomplete distal RTA into those that have a urine pH above 5.3, 5.5, and those that can still acidify the urine. So this is a funny group, because how could you have normal acid base status, the ability to acidify, and you're still calling it incomplete RTA? Well, it was found that some of these patients have decreased ammonium excretion. So this incomplete RTA characterization is not based on the inability to acidify, but based on the ability to excrete the appropriate amount of ammonium when you acidify the urine. So that's another subtlety that has been described. As far as those that can't acidify below 5.3 or 5.5, they're also broken down by their ammonium excretory capability. So this is a new sort of sub-categorization, or breaking down what happens to the urine pH even further, which has come into play in the last 3 to 4 years or so. you have to look at not only the urine pH, but the ability to excrete ammonia. So this is sort of the interesting group. Conceptually, they have normal blood gases. They can acidify the urine normally, but they can't excrete the ammonia normally. And they're felt maybe to have mutations in the Rh proteins in the collecting duct. This is a hypothesis. The UGCG is an enzyme involved in glycosphingolipid metabolism. And it's now felt that these are important in the interstitium of the kidney outside the tubules to bind ammonia and then allow it to be transported into the collecting duct. There's some papers written on this. So mutations in these proteins in mice can actually cause a distal RTA-like phenomenology. It's not been described in man yet. So when you hear about incomplete RTA or incomplete distal RTA, you're talking about someone who you wouldn't normally pick up, who has normal blood gases, normal acid-base status systemically, but that shouldn't make you think they don't have a problem with the physiology of the collecting duct, either in the absolute ability to acidify the urine or with normal ability to acidify, but don't get their ammonia excretion rate up appropriately. These are things we tend not to think about clinically, but the thought is that if you pick these things up earlier, you don't have to wait till they have full-blown systemic acid-base abnormalities. Now, when you get a metabolic acidosis outside the kidney, for instance, in lactic acidosis or ketoacidosis, there's a bunch of things that happens. The kidney responds to this. First, in the proximal tubule, there's increased ammonia production and new renal bicarbonate generation from glutamine. You make a lot more alpha-ketoglutarate and convert the alpha-ketoglutarate to bicarbonate. Mentioned here is gluconeogenesis. I didn't mention that earlier, but the alpha-ketoglutarate is turned into bicarbonate not only in the Krebs cycle, but if there is gluconeogenesis occurring, it can also generate new bicarbonate via gluconeogenesis. But this really should say the Krebs cycle predominantly and some gluconeogenesis. So that occurs, and that's how the kidney tries to compensate for the lactic acidosis. And it is occurring. And you only get a drop in your systemic pH when this can't compensate for the amount of lactic acid being made. It's only when it exceeds the increased production of bicarbonate from new bicarbonate that you start dropping your systemic pH or stop dropping your blood bicarbonate. There's also a change not only in new bicarbonate production, but in the urine to renal vein ammonia partitioning. I said before, 60% of the ammonia ends up in the urine, freeing that amount of new bicarbonate to match the hepatic proton load. But if I significantly increase the proton load, not from the liver, but from lactic acid or from ketones, the kidney actually recognizes this and puts a much greater percent of the ammonia it's making from the alpha-ketoglutarate reaction into the urine. And that makes sense because now you've freed up even more new bicarbonate than you would have otherwise. And that's why you get more ammonia in the urine in extrarenal metabolic acidosis. Not only are you making more, but of what you make, more of that is going into the urine as a compensation to free up more of that new bicarbonate to match the proton load coming with the lactate. You also increase HPO4 going to H2PO4, or so-called titratable acid excretion, because the proton secretion by the proximal tubule and the collecting duct, which is required to change the HPO4 to H2PO4, is increased. There's also decreased ureagenesis in the liver. That's not well known. You make less urea in metabolic acidosis, and that makes sense because then you're consuming less of the bicarbonate that's coming into the liver to make urea. And these two processes are defective in distal RTA. So if someone says to you, why do I get a metabolic acidosis in distal RTA? There's two reasons. One, this enhanced partitioning of sending more of the ammonia into the urine versus the renal vein cannot occur, and I'll show why in a minute. And secondly, you don't make as much H2PO4 from HPO4 because the H secretion is defective. So that's easy to understand. But what's going on here? What's going on here is you impair the ability of NH4+ to get into the urine. And so more of the ammonia now that's made in the proximal tubule from in the alpha ketoglutarate reaction is going to the renal vein, and that's just using up inappropriately more of this bicarbonate that's been made, new bicarbonate. The reason you can't get the ammonia into the urine is because you have to be able to acidify the urine to trap the ammonia in the urine, convert that NH3 that enters to NH4 plus. It's not a problem with the RH proteins. The NH3 can get in, but it's not converted to NH4 plus. And unless it's converted to NH4 plus, it doesn't continually enter. It continually enters because every time it enters, it's converted to an NH4 plus, which stays in the lumen and comes out in the urine. But if you don't have this conversion to NH4 plus, you don't have the NH3 continually entering because you don't have the NH3 gradient. Every time you make NH4, you lower the NH3 in the lumen here, and you have a constant gradient for NH3 to passively go through the RH proteins into the lumen. But if you don't do this, then you lose that gradient and NH3 doesn't enter as much. And now you just have much more NH3 or NH4 plus going to the renal vein. It can't come out in the urine. And if you measured the ammonia partitioning in someone with distal RTA, which has never been done, but if you did, you would find out that there's a much greater fraction of the ammonia that the whole kidney is producing going into the renal vein rather than the urine. And again, the easier one to understand is that you just don't secrete protons normally, either because you have a B1 subunit a defect or the A4 subunit, or bicarbonate can't get out on AE1, as I showed. All these things stop the acidification, and you just don't convert as much HPO4 to H2PO4, and therefore you don't generate as much new bicarbonate. So as far as the abnormal urine to renal vein ammonia partitioning, that's an explanation, but not something we can actually measure clinically, because we can't measure the renal vein ammonia to know for sure, but we can measure that there's less ammonia in the urine. We can never know when we see that, by the way, whether that's due to decreased generation or due to or an abnormal partitioning. It could be one or the other or both. Remember, there's an X amount made by the kidney, and then it gets split 60-40. You can't tell if the total production is down because we can't measure the renal vein plus urine total. You don't have access to the renal vein. And therefore, we can't measure the partitioning either. All we can measure is the amount in the urine. And you have to make an inference as to whether that's due to decreased production or abnormal partitioning, which means that more of the ammonia that was made is going into the renal vein rather than the urine. So to measure the urine ammonium at UCLA now, as I said a few times, we don't have to do these things anymore. We can measure the ammonia directly. We can send it to the lab, and we're lucky in that sense because there's only a handful, if less than that, hospitals in the country that can do it. I think ours and maybe one other. So you're fortunate in that sense that you don't have to use these indirect methodologies to do it, which are inaccurate. You need to know them for an exam, but they're really historic, and hopefully one day we won't see these terms anymore as far as assessing urine ammonia. But there is a clue that there is enhanced ammonium going to the renal vein, which very few nephrologists are aware of, and that is that a number of papers now have described patients with distal RTA who have a high blood ammonia level, and that is really not something you'll hear any attending tell you or any other fellow in any hospital. But if you look at the literature, it's very clear that the blood ammonia level is higher in these patients. And that makes sense if this mechanism is right, that you have less ammonia coming into the urine, more going into the blood. And remember, there's also less ureagenesis. There's less ammonia consumption in the liver when you have any metabolic acidosis. So they have less ammonia conversion to urea. They have enhanced renal vein ammonia delivery to the blood, and that is what's causing the elevated ammonia. So it's something you could do. It's not done clinically, but if you do suspect that someone had untreated, we're talking about, because once you treat, you correct the metabolic acidosis, you change the renal vein urine ammonia partitioning. But talking about a de novo patient, if you have someone who you think has de novo distal RTA, it would be interesting to measure the urine, and see that it's low, and the blood and see that it's high. And in the presence of a non-gap metabolic acidosis with hypokalemia, there's nothing else that's going to do that other than distal RTA. So it's an interesting experiment of nature to try. And conceptually, it's nice to try just to see whether it holds in your patient. But a number of papers have shown it already. Now, another question in these patients is, why do they have these other electrolyte abnormalities? Why would they get hypercalciuria? Well, if you ask 100 nephrologists, they'll say, well, it's the metabolic acidosis. And that's not true. There are many other causes of metabolic acidosis where you don't have hypercalciuria. So it's not that the calcium is coming from bone. And actually, years and years ago, in 1982, Soriano and others showed that it's a problem with calcium absorption by the nephron, not increased calcium coming to the glomeruli that you're filtering. So try to rid your mind of the thought that the hypercalciuria, the nephrocalcinosis, the calcium phosphate stones are due to bone resorption. That's not the mechanism. And we'll talk about it in more detail in a minute. Now, in fact, it was shown, this just shows the nephrocalcinosis, how the ducts of Bellini here, these are the terminal portions of the intermedullary collecting duct. They're filled with calcium here. This is a stain for calcium, and they're black. This is typically what you see when you stain for calcium in these kidneys. What was shown years and years ago is as you correct the bicarbonate, you actually decrease the calcium excretion. So it does seem to be blood pH or bicarbonate dependent, but it's not due to the fact that you have less and less bone resorption. As far as the treatment, we give K-citrate with or without thiazides to treat this. Try to get the blood and this is specifically the hypercalciuria, not the metabolic acidosis. These are the goals of treating the hypercalciuria. You get the bicarb to about 22. That will help normalize the calcium excretion. You also try to normalize the citrate excretion because you're trying to get the citrate up in the urine, and that will help get rid of the ionized calcium that's binding to the phosphate. So it'll decrease the propensity to get calcium phosphate stones. And you give thiazide if the urine pH gets above a certain amount to prevent the alkalinization of the urine from the citrate, because what you don't want to do is alkalinize the urine too much, which you will do. Remember, citrate has carboxyl groups, just like alpha-ketoglutarate, just like lactate, and it's converted into bicarbonate. And that's what you're doing when you're giving the citrate. But you also can end up getting too much bicarbonate in the urine, raising the urine pH, and that will lead to more calcium phosphate precipitation, which is exactly what you don't want to do. So once the urine pH gets above a certain amount, you then give thiazides to help try to stimulate calcium absorption instead of pushing the citrate. So this is the mechanism of the hypercalciuria. If anybody asks you, it's the TRPV5 calcium channel that's present in a portion of the nephron called the late distal convoluted tubule. It's the portion right after the cortical thick ascending limb in the cortex. There's two portions to this. This is the second portion. It's also present in the next part of the nephron called the connecting tubule. So it's expressed on the apical membrane, and it's responsible for taking calcium from the lumen into the cell, but it's very pH sensitive. And this is an experiment in animals where if you just give ammonium chloride and acidify the blood of the animal, you can see that the expression of this thing, here it is in the control animals, the green is the expression in the distal convoluted tubule. But in a metabolic acidosis, the cell just doesn't make as much. It turns out also that this channel is very pH sensitive, and so its transport of calcium actually also decreases when you acidify the pH in the cell or the outside of the channel. So there's sort of many mechanisms why it doesn't transport calcium normally. One, it's pH sensitive in and out, and also its expression decreases. And this is just an example here in a normal animal when you give ammonium chloride or acetazolamide, you get a lot more calcium excretion. This is a TRPV5 knockout mouse missing the transporter. First, note how much calcium is coming out of the urine, 300 micromoles a day instead of 25. So that just alone shows you how important it is for preventing hypercalciuria. But the interesting point is that without that transporter, the animal is not sensitive to metabolic acidosis anymore, even though it's got the same bones as the wild type animal. So this proves that it has nothing to do with the bones. It's solely due to the presence of this transporter, or this channel. Sorry, it's not a transporter. This channel being present. And when you don't have it, your body no longer responds to metabolic acidosis by getting hypercalciuria. Now, this wasn't done in humans, it's done in mice, but presumably we have the same similar physiology. So the pH, or the metabolic acidosis causing of hypercalciuria, requires the presence of a normal TRPV5 channel in the DCT tube, connecting tubule area of the nephron. So this is a very interesting and important experiment. The other thing that happens in metabolic acidosis or in hypercalciuria, why when we get hypercalciuria do we normally not get nephrocalcinosis or calculi? Because you have so many patients with hypercalciuria who don't get renal stones or nephrocalcinosis. And one of the thoughts is when you get hypercalciuria in the principal cells and in the intermedullary collecting duct cells in the nephron, the calcium binds to the calcium sensing receptor. Remember, there's a calcium sensing receptor in our parathyroid glands that we treat pharmacologically, but it's also present throughout the body in different cell types, including the nephron, but it has a different role. It's felt that when calcium binds to it in these cells, aquaporin 2 actually can't insert in the membrane and you get polyuria. That's one of the mechanisms why patients with hypercalciuria get a nephrogenic diabetes insipidus, is because when calcium binds in the lumen here to the calcium sensing receptor on these cells, you can't absorb water normally. So you get a nephrogenic DI. Aquaporin 2 just doesn't insert in the membrane, but that's good in a certain sense teleologically, because it dilutes the calcium concentration and you don't end up having as much of a chance of getting calcium stones. The other thing that happens is it actually stimulates urine acidification in the type A intercalated cell. It's one of the factors that is felt to stimulate proton secretion. Mechanism not clear. And that's good too, because that prevents calcium phosphate from coming out of solution. So not only do you dilute the urine when you have hypercalciuria, but you actually stimulate urine acidification. But patients who have an abnormal vacuole or proton pump or AE1 mutations on the basolateral membrane cannot acidify normally. So they lose this mechanism that we all have to prevent calcium phosphate stone formation when we have hypercalciuria. The mechanism of hypokalemia, I can just start off by saying I'm going to show a lot of things here, but it's just a lot of jargon. Probably none of these is true, but people have hypothesized, and they can get very severe metabolic, or they can get very severe hypokalemia. I've had patients with distal RTA with Sjogren's syndrome who have potassiums of 1.7. I mean, it can get very, very low. And it's been attributed to proximal tubule decreased K transport. I'm just showing these. You don't really have to get into these in details. I have the references if you're interested. The thick ascending limb, decreased K absorption via the ROM-K channel. All these are invoked in the literature. They may give you this on an exam. I doubt it. Again, when you see many explanations in medicine, you know that likely none of them are true, or there's a lot more going on. Okay, and lastly, how do you diagnose distal RTA? Well, the current thinking is that the easiest way, and this has gone through a lot of different, a lot of evolution. I actually have these in the reverse order as far as temporally. What we used to do was give a sodium bicarb load. And what you measure is urine blood gases. And if you find that in a normal person, the PCO, when you do this, you find that the PCO2 in the urine goes up. And we'll talk about why that occurs another time. Just for now, take it as faith that if I give you this amount of bicarbonate and then measure your urine blood gases, the PCO2 in your urine is going to go up to 70 or 80. So it's usually going up to a level above 30 higher than your blood. And if you see that, you know the person does not have a distal RTA. We don't do this. In fact, we never used to do this a lot just because it was such a pain to do. You have to collect the urine under mineral oil to prevent the carbon dioxide from being excreted. You have to keep measuring it till you get this up to this level, and you don't really know when to measure it. You sort of guess. So it was really inconvenient. What should be done now is this easier test. It's known also, and we'll talk about it another time, it's somewhat controversial, but it is known that if I give you Lasix, your urine becomes acidic. And that's not, well recognized by most nephrologists. But it's another way of acidifying the urine without having to give ammonium chloride like what was done historically. So it's a very easy way of acidifying the urine. You give this amount of Lasix, and what you do is you start measuring the urine pH, and by the 4th hour in all of us, the pH should go below 5.3. If it does that, then that tells you the patient does not have hypokalemic distal RTA. Now, they could have incomplete RTA with decreased ammonia excretion. So some people also measure the amount of ammonia in the urine. By the 2nd hour, that's how much ammonia should be coming out per minute. You measure it over a few time points. And again, given that we can measure ammonia now at UCLA, this is something that could be done. We do this in addition to the pH, because remember, there's some people who have incomplete distal RTA who can acidify, but whose ammonia excretion rates are below normal. Perhaps they have Rh protein mutations in the collecting duct, so the NH3 can't get from the outside the tubule into the lumen of the tubule normally. Unknown, just a conjecture. So this is easy to do, and the only other subtlety is, remember, there's certain types of hyperkalemic distal RTA, like post-obstruction patients where they get a hyperkalemic distal RTA. It's also called a voltage type of distal RTA. Nothing to do with hyporenin, hypoaldo. They can acidify the urine normally, even though they have distal RTA. We'll talk about that another time. But if it's hypokalemic distal RTA or in a normal person, you should be able to get the urine pH below 5.3, and someone with hypokalemic distal RTAs that we talked about, like the vacuolar proton pump subunit patients or the AE1 subunit patients, they don't acidify normally. So it's a nice way of doing it, and you can know objectively on the chart that the patient must have distal RTA without guessing. The urine ammonium is not always the best to do, even though it's one of the tests people talk about in diagnosing distal RTA, just because the urine ammonia excretion may be below because of a production problem. It may be a problem with glutamine uptake. And for instance, if you have someone with CKD, they also have a low ammonia excretion in the urine, but it has nothing to do with acidifying the urine. They acidify normally. The problem they have is they don't metabolize glutamine normally. They don't make enough new bicarbonate in that reaction. That's why patients with CKD get a metabolic acidosis. It's a problem with new bicarbonate generation, and therefore they also don't make ammonia in that reaction. That's why their ammonia is low, but it has nothing to do with distal RTA or a proton secretion problem in the collecting duct. So a low urine ammonium, you never know, as I said before, is it due to a production problem or a partitioning problem? And here we're talking about a partitioning problem. So when you can't acidify the collecting duct normally, you have inappropriate amounts of the ammonia you make going back into the renal vein rather than the urine. Okay, so I think we'll stop there today. And does anyone have any questions about what I talked about? I know we went a little bit longer. Okay, have a great night, everybody. Thanks for joining.